Characteristics of mold. Let's see about fungi. Fungi are the group of eukaryotic microorganisms. Fungi are divided into two groups: multicellular filamentous mold and unicellular non-filamentous yeast. In this video, we are going to discuss about characteristics of mold. Introduction. Molds are eukaryotic multicellular filamentous fungi. Growth of mold appear as fuzzy cottony appearance. Some are colored due to pigment production, some are colorless. Some molds may be harmful, involved in the spoilage of foods, produce mycotoxins and the contaminated food items. Some are beneficial, involved in the manufacture of food items, produce antibiotics, enzymes, organic acids. Special molds are useful in the manufacture of certain foods or act as ingredients of foods. Let's see the characteristics of mold in three different aspects: morphological, cultural, and physiological. Moving on to morphological characteristics. The form and structure of mold as judged by their macroscopic and microscopic appearance used in their identification and classification of mold. Hyphae and mycelium. The mold thallus consists of mass of branching intertwined filaments called hyphae, and the whole mass of these hyphae known as mycelium. The hyphae may be submerged and aerial. Submerged hyphae growing within the food, aerial hyphae growing into the air above the food. The hyphae may also be classified as vegetative and fertile hyphae. Vegetative hyphae involved in the distribution of nutrients, water. Fertile hyphae involved in the production of reproductive parts. In most of the molds, fertile hyphae are aerial. In some molds, they may be submerged. Sclerotia, tightly packed masses of modified hyphae. It provides more resistance to heat and other adverse environmental conditions. Microscopic examination of hyphae reveals characteristics useful in the identification of genera. Molds hyphae divided into two groups: septate and aseptate hyphae. Septate hyphae, cross walls dividing the hyphae into cells. Non-sinusoidal aseptate, consisting of cylinders without cross walls. The non-septate hyphae have nuclei scattered throughout their length and considered multicellular. The hyphae of molds are clear, but some are dark or smoky. Hyphae may appear uncolored and transparent on microscopic examination, but colored when large masses of hyphae are viewed macroscopically. Reproductive structures. Those fungi producing sexual spores known as perfect fungi, and those do not have sexual phase known as imperfect fungi. Example for sexual spores: oospores, zygospores, ascospores, basidiospores. Asexual spores such as conidiospores, sporangiospores, arthrospores, chlamydospores, and blastospores. Asexual spores. Conidiospores. Conidia are cut off or bird from fertile hyphae. Conidiospores arranged in chains at the end of conidiopore. It is produced by Aspergillus and Penicillium. Sporangiospores. Sporangia have sac-like structure at the tip of fertile hyphae. Sporangiospores found within sporangium. It is produced by Mucor and Rhizopus. Diagrammatic difference between the arrangement of sporangiospore and conidiospore. At the end of the sporangiopore, sporangium is present. Within the sporangium, sporangiospores are there. In case of conidiospore, at the end of conidiopore, conidiospores arranged in a chain-like appearance. Sporangiospores found within the sporangium, conidiospores arranged at the end of conidiopore. Arthrospores. Fragmentation of hyphae, cells become arthrospores. It is produced by geotrichum. Chlamydospores. Mycelium stores up reserve food, swells and form thick wall than that of surrounding cells. Chlamydospores found within the hyphae. Blastospores formed from buds of the parent cell. Sexual spores. Oospores. It is produced by non-septate mold, for example picomycetes and oomycetes. It is formed by the union of small male gamete and large female gamete. Zygospores. It is formed by the union of tips of two hyphae produced by zygomycete. Ascospores. Formed after the union of two cells from the same mycelium, where ascus are formed. There is sac-like structure. Usually it contains 8 spores. It is produced by ascomycetes. Basidiospores. It is produced by basidiomycetes, for example mushroom, plant rest and smut. Specialized structures. Special mycelial structure or pods helpful in the identification of mold. Columella. Swollen tip of sporangiopore usually projects into the sporangium. Apophysis. Swelling below the sporangium. Stolon. Horizontal hyphae connects sporangiopore. Rhizod. Short thin filament root-like structure. Sterigmata or phyalids. It's a flask-shaped structure that contains conidia, food cell, basal cell of conidiopore. Moving on to cultural characteristics. Saprodes dextrose agar and potato dextrose agar used as a selective media for the cultivation of mold. The growth of mold appear as loose and fluffy, others are compact. Some looks velvety on the upper surface, some dry and powdery, others are wet or gelatinous. Some are pigmented, for example red, purple, yellow, grey, black, green, blue-green, orange, pink, lavender, brown. The appearance of reverse side of a mold on an agar plate may be striking, like opalescent. The pigments in the mycelium are the characteristics of masses of asexual spores. Moving on to physiological characteristics. Moisture requirement. It requires less moisture when compared to yeast and bacteria. Temperature. It is mesophilic. Temperature vary from 25 to 30 degrees Celsius, but some grow at 35 to 37 degrees Celsius. For example, Aspergillus species, some are psychrotropic. They grow well at refrigeration temperature. Some are thermophilic. It grows more than 45 degrees Celsius. Molds are aerobic. pH requirement. It may vary from 2 to 8.5, but most of them grow at acidic pH. Food requirements. Molds utilize from simple to complex substances because it possesses various hydrolytic enzymes such as amylase, pectinase, lipase and proteinase, helpful for the breakdowning of the substances and make them available for the growth of microorganisms. Inhibitors. Compounds inhibitory to other organisms are produced by some molds, such as penicillin produced by Penicillium chrysogenum and clavacin produced by Aspergillus clavatus. Some compounds are mycostatic, inhibiting the growth of mold, like sorbic acid, propionic acid, acetic acid. Some molds are fungicidal, killing other molds. Thank you for watching.